Now we talk about the membrane proteins. So those are membrane proteins. They are responsible for different cellular activity, for example, signaling, transport, sensing, structure support, and so on. Okay, so it depends on the way they incorporate to the membranes. There are three classes of the membrane protein. One is integrate membrane protein, or we call it transmembrane proteins. Okay, they will fully penetrate the hydrophobic core. Okay, and the second will be lipid anchor protein. Basically, several lipids will directly conjugate to the amino acids, and those lipid anchor protein will use those lipids to incorporate to the lipid bilayers. Okay, and the third is a peripheral membrane protein. They will bind to the membrane indirectly. They bind to the other component or bind to the other membrane protein or other membrane stuff. Okay, so first, how those integrate membrane protein or transmembrane protein localize in this membrane. They also need to deal with this hydrophobic core. Okay, so basically the a protein is amino acid sequence, right? So it's come with the N terminal and a lot of amino acid and all the way to the C terminals. So they will arrange those transmembrane protein. They will arrange their amino acid to kind of hydrophilic. They put a hydrophilic amino acid here. Then conjugate, then come with the hydrophobic amino acid, all the way from this side to the other side of the lipid. Okay, they basically form the alpha helix, which is a highly hydrophobic secondary structures. Then outside the lipid bilayer, they will put a lot of hydrophilic amino acids. Then go back hydrophobic, hydrophilic, hydrophobic, hydrophilic, hydrophobic, hydrophilic. They use this way to concentrate the arrange their hydrophobic amino acid to the center and help them to embed to this hydrophobic layer. Okay, so if you check their amino acid sequence, you will see such a pattern, which is a typical transmembrane protein pattern. It's a non-polar amino acid, which is a hydrophilic, hydrophobic, hydrophilic, hydrophobic, hydrophilic, hydrophobic, hydrophilic, hydrophobic. And we can kind of easily predict those parts is the amino acid region penetrate the membrane. We call this a transmembrane domain. Okay, yeah. Okay, so if you check those amino acid sequences, you will know the alpha helix mainly composed of those hydrophobic amino acids shown in the green and the yellow color. Of course, there are few hydrophilic amino acids, but most of them will be hydrophobic. Otherwise, they cannot form such a highly hydrophobic secondary structures. And some software, online software, can easily predict their level of hydrophobicity. So we call this hydrophobicity index. Okay. So in this case, it will be those amino acids from the first amino acid to the 100 and maybe 10 amino acid will be low hydrophilic here in this region and highly hydrophobic here, and then hydrophilic here. Then we will predict it's likely this protein have only one transmembrane domain, okay? And actually, usually those transmembrane domain contain 20 to 25 amino acids, usually, okay? It depends on the angle and what kind of way they incorporate to the membrane. So, yeah, and for those, the example B, bacterial rhodopsin, it contains a lot of different 1, 2, 3, 4, 5, 6, 7 domain region contain a highly hydrophobic amino acid. Okay, then we will predict this protein may have a 7 transmembrane domain. Okay, likely you still need the kind of cryo-EM or other structure analysis to know the answer, but based on the amino acid, it's likely energetically favorable condition they will have a 7 transmembrane domain. Okay, in some cases it's difficult to tell. Okay, I will tell you the one example. For example, sometimes those alpha helix, they will kind of go this way and then go out based on they will kind of form such a beta strand or other non-alpha helix structure, then go back here. Okay, so based on the amino acid, they will be kind of really short alpha helix structure. And you will kind of ignore or think this is not a transmembrane domain, but the truth is not. They still the transmembrane domain, they just go away. They are not the entire, fully extend the kind of composed of the 20 to 25. It maybe only contain 10 amino acids. Okay, so we still need the structural biology analysis. Okay, so then textbook, when you read the textbook, it just gives you a lot of example that waste your time. Okay, but for the course, I don't only take 10 seconds to finish that part. You may read a lot of 3 paragraphs, then you only get one information. They just give you an example. This is a single transmembrane domain protein. That's it. Okay, so it's a glycoprotein A, which is this protein only contains one transmembrane domain. And this will be the example for the multiple transmembrane domain protein. It's a bacterial rhodopsin. You will see a lot of multiple domain. That's it. And in some cases, you just cannot really predict how many transmembrane domain they have, because they sometimes will use the half alpha helix, then go away, then come back with the other alpha helix to kind of achieve their chemical property to fit in this chemical property. Okay. And this will be the also kind of example. No matter what, the protein need to embed into the membrane. They need to fit in this hydrophobic environment. Okay, but may have some kind of special case like aquaporin. Aquaporin is also the transmembrane protein. Okay, but aquaporin is the kind of key protein allow the water molecule to go across the. They have a kind of tunnel inside their protein, okay, and it can allow the water molecule to go through that tunnel. So this is the stimulation movie. The yellow dots are the polar head of the lipid. The green are the fatty acid tail. Yellow dot will be the other side of the polar head. And the blue will be the alpha helix of the aquaporin, which is one kind of transmembrane protein. And the red will be the water molecule. Water is not, it's hydrophilic. Okay, they are water, they are not hydrophobic. They actually cannot kind of freely penetrate the hydrophobic lipid bilayer. But aquaporin provide the tunnel inside their proteins, which is a really tiny water-variable hydrophilic environment inside their protein, and a little bit open to this end and to the other end. So those water molecules can go through the aquaporin between these two sides. Okay, but the surface of the aquaporin is still hydrophobic. The tunnel is hydrophilic. That would be the inside, but the surface of the aquaporin is still hydrophobic. So this white cation, this stuff is aquaporin protein. So the surface is still hydrophobic, so they can align with a lot of hydrophobic lipid. As you can see here, those are fatty acid tail. It's a cation to simulate their situation. Okay. We will introduce more later. Okay. So you may know there are some common structure for the transmembrane domain. The first is most of them are hydrophobic amino acid, and most of them are alpha helix. Okay, but it always have some exception in biology. So not all amino acids in the transmembrane domain are non-polar amino acids. For example, in this T cell receptors. So this is the surface membrane, a cell membrane of the T cell. When the cell need to activate certain immune activity, they need to kind of activate T cell. To activate T cell, you need to assemble the T cell receptor on the surface of the T cell. So those are different. In the steady state, they don't need to assemble each other, but when cell need to activate the immune response, they need to assemble with each other. So of course, there are a lot of, those are transmembrane protein. They're still mainly based on the alpha helix, but none of all amino acids in the transmembrane domain are non-polar amino acids. Some of them have a charge. They are polar amino acids. So those are positive charge and negative charge. During the assembly, they can help them assemble and stabilize those T cell cluster. Okay, and activate the downstream immune response. Okay, through those electrostatic force. Okay, and also one exception: not all transmembrane domain are alpha helix. Some of them can be the beta strand, which is the other common kind of secondary structure. Usually, we will consider beta strand would be the hydrophilic, but in this case, they still those is a bit. I think what is this protein? It's a kind of porine protein structures, and this is a membrane. They are mainly based on beta strand protein, but they will put a lot of hydrophobic amino acid in these regions. So it kind of put a lot of hydrophobic amino acids on the surface of the beta strand. So still keep overall structure hydrophobic, okay? And next, just give you, textbook just give you, yes, it's not the special case. There will be a lot of different protein have used a beta strand to embed in the membrane, but they will put a lot of hydrophobic amino acid to the surface of those beta strand, but still keep the structure of the beta strand. To achieve the enough hydrophobicity, then they can stably embed in the membrane, okay? So it will be the two inception. Okay, so next we jump to the lipid anchor protein. So lipid anchor protein, there are some modification. So many enzymes inside the cell, they can conjugate lipid to the amino acid, okay? Especially in the Golgi, okay? So the Golgi or some organelle, they just synthesize some lipid and conjugate directly to, for example, this is glycine, cysteine, okay, to this protein, to the N-terminal or C-terminal tail, or put the more complicated lipid here, we call the GPI anchors to these proteins. Okay, so we call this acetylation, prenylation, and the GPI anchors. So they will conjugate to the different side of the protein and the different side of the cell. So for example, the acetylation, they will conjugate to the cytosolic leaflet and to the N-terminal cytosolic leaflet and to the N-terminal of the protein. Okay, it depends on the enzyme. The enzyme will recognize the N-terminal and the enzyme localized to the cytosol pool. Then they can do this acetylation, okay? The enzyme will recognize the C-terminal tail of the amino acid and localize in the cytosol, they will do the prenylation, okay? And the enzyme localized to the Golgi lumen, okay? As I mentioned before, the orientation will be conserved during such a protein secretory or vesicle delivery, right? This site, exoplasmic leaflet, always face to the extracellular space or to the lumen of the organelle, okay? And the enzyme, so this protein inside the cell, during the synthesis, they face to the Golgi lumen, okay? And many enzymes in the Golgi lumen, they can synthesize, they'll put a lot of such a modification in the Golgi lumen, then they gradually move to the surface and kind of budding out. That is why this protein will face to the extracellular space. Okay, such orientation has been conserved during the synthesis. Okay, so this is GPI anchors. It's exoplasmic leaflet and it's conjugate to this C-terminal. And they will know a lot of surface protein, they has been conjugated with a lot of carbohydrate or carbon, sugar. Okay, a lot of carbohydrate chain will link to the serine surrounding and the asparagine on the surface of the protein. Why the surface protein? Because the enzyme required to conjugating those carbon to those amino acids are all located to the Golgi lumen. Okay, so during the protein synthesis, they will gradually move to the Golgi. So in the Golgi lumen, there will be a lot of modification enzyme. They will put the sugar to those specific amino acid. Then this protein, after maturation, they finish all modification, they will gradually move to the plasma and budding out, then they face to the extracellular space. Okay? Yeah. So that is why almost all carbohydrate or glycoprotein, okay, are the surface protein because most of the enzyme localized to the Golgi lumen. Okay? And those sugar are really important because it's really important for the cell to recognize the environment or the cell to recognize other people. We can recognize other people to recognize other cell. Okay, we can recognize other people based on their face, but the cell can recognize what kind of cell type. For example, the immune cell, they should recognize what's our body cell, what's our kind of tumor cell. Okay, it's based on the surface protein, and those surface protein will modify with a different carbohydrate. They basically just recognize those are sugar and the lipid. Okay, so just give you an example. The human ABO blood glucose antigen. So those are a lot of different kind of sugar. So those are sugar. This is a red blood cell. Okay, this like shape. A lot of membrane protein on this red blood cell. Those sugar are conjugate here. Okay, you need to know what I'm talking about. Okay, this part would be this. We call the O antigen. Okay, those are really basic sugar complex. We call it O antigen. And the people whose red blood cell only contain those O antigen, we call the type O blood people. Okay? But if the people, the human contain the gene which can encode those S-galactosamine transferase, they will conjugate one more this sugar. We define this A antigen. The type B person who carry the gene which encode this transferase, galactose transferase, they will conjugate one more galactose to those basic units. We call this a B antigen. Okay? And the people have both of these two genes. They can generate this and also generate this on their red blood cell. We call them type AB a person. Okay? Yeah. So type A blood peoples have A antigen on the red blood cells, but they have anti-B antibody in the serum. Okay. So the what does that anti-B antibody means? It means they will recognize B antigen and form really strong interaction. Okay. So the anti-A cannot incubate with the red blood cell with the A antigen. Otherwise, they will form really strong interaction and form the cluster. Okay, and then your blood cannot freely move, then you die. Okay, so the red blood cell, the antibody in your blood vessel, blood flow, those are red cell, right? And the solution we call the serum. 血清, this is a red blood cell, it's a cell. Okay, a lot of blood cell, immune cell, and the non-cell solution we call serum. And there will be a lot of antibody here. Okay, so if this A antibody, anti-A antibody, you don't put those anti-antibody to the cell with the A antigen. They will interact with each other and form the cluster and form the kind of thrombus, then kill yourself. Okay, so that is why the red blood type cell, they don't have any antigen on the red blood cell, but they have both anti-A antibody and anti-B antibody in their serum. They cannot receive the blood from those person, right? Because they have anti-B antibody, they will recognize this and form the clock, and form the clock. So they can only receive the blood type would be the O. But the AB guys, they don't have any antibody in their serums. They can receive any blood type. And the B type person, they have anti-A antibody. They cannot receive the blood from B or O. It will be the same kind of concept, okay, in this case. Okay, so that would be the example of textbook teachers for the lipid anchor protein. Then we use this example to show you how peripheral protein bind to the membranes. So we use the example is a phospholipids. Okay, so phospholipids means the enzyme digests phospholipid. So we call S phospholipids. Okay, so this is the chemical structure of the polar head of the phospholipid. So their phospholipid, lipase A1, A2, and C and D, they are enzyme will kind of break down the chemical bond of those polar head respectively. So why they need to break down those phospholipid? Because sometimes they need to do the homeostasis, they need to modify, or sometimes phospholipid may damage. They need to kind of destroy them for reuse. Okay, you just need to use such a feedback mechanism to clean up or to do some. Sometimes lipid may generate too much, so the cell need to digest them for the reuse or better use. Okay, but to digest for the enzyme digestion, the enzyme need to directly contact with the substrate. They cannot digest the substrate in the long distance. Okay, there's no way. They need to directly contact them and digest those chemical bond. Okay, so how those phospholipase will bind to those phospholipid polar head and digest them? So they have a really elegant design. So this is a cartoon of the phospholipase A2. They have a tunnel inside. Okay, and in the entry of those tunnel, they put a lot of polar ring, which is hydrophilic, and it will help them to kind of roughly bind to the polar head, and a little bit suck out those phospholipid. Okay, if they if they have some structure damage, they kind of cannot form the really strong interaction with other lipid. They will be easily sucked out by those phospholipase. Okay, then once it suck up to attract those lipids into the tunnel, the wall of those tunnel are composed of a lot of non-polar amino acid. That is why the fatty acid will be kind of really enjoy the alignment with the wall of the tunnel. Okay, then they put the calcium. There is a calcium molecule in the end of those tunnels, and those calcium will bind to the polar head. So they have a hydrophilic molecule to retain this phospholipid, and they have a lot of hydrophobic amino acids on the wall of those tunnels will also keep them there. Then they can have a lot of time and a good environment to digest this bond. Okay, that is why they can disrupt this and then go away. Okay, so it just gives you the example of how peripheral protein, they may just through some hydrophobicity or a certain really elegant design of their protein structure to do their job, to bind to the membrane and then cleavage those polar head. Okay. So the one more thing you need to know is the detergent. Okay, so detergent are just basically chemicals. Okay, but those chemicals are all amphipathic. It means those are chemicals with strong hydrophobic and hydrophilic. So the yellow part will be the hydrophobic part of those 4 different detergents, and the green will be the hydrophilic part. Okay, so this detergent can easily disrupt the membrane, really easy. Okay, for example, this is a cell, one kind of cell. You just add a detergent. Just add detergent right now, the membrane disrupt. Okay, so in some case, we need to kind of extract the membrane protein for the study, or sometimes we need to kind of damage the membrane, break down the membrane, we will add the detergent. And that is also why we can kill the bacteria or kill the virus when we add a detergent during the hand wash. Okay, yeah. So why this can disrupt membrane? Because they are amphipathic. Hydrophilic will align to the hydrophilic part of the membrane. Hydrophobic will align to the hydrophobic part of the membrane, but they look really different to those lipid protein, right? So they just align to the really ugly structure or based on their structure. That is why finally they cannot maintain those lipid bilayer and disrupt the whole membrane. Okay. But those detergent have 2 major groups. One is ionic detergent. The second is non-ionic detergent. So ionic detergent is mean they have a charge. Non-ionic detergent, they don't have charge, they are neutral. Okay. What's the manner to think about the ionic or non-ionic? Usually when you do a lot of biological experiment, you need to deal with the protein. You need to kind of extract your membrane protein, collect your membrane protein for the following experiment, okay? All of them, detergent can disrupt the membrane, can help to extract the protein. That's no problem. But ionic detergent, they have a charge. Once you extract those protein, those charge will bind to your amino acid and totally denature your amino acid. For example, our protein, they can contain the enzyme activity or can be still functional if they can form the proper 3D structure. Okay? But if you bind to a lot of charged molecule, they can kind of align them to the kind of linear form. We call this denature. Okay? Then they're still there, but they don't have any enzyme activity. But those are good for certain experiment if you don't care about the enzyme activity. For example, the Western blot. You just care about the protein label. You just collect them and put, for example, one kind of ionic detergent, SDS. They can denature your sample and collect the membrane protein and denature them. So they don't keep their original structure. They don't have enzyme activity because you don't care about their enzyme activity. You care about their amount, right? So that is why you run the SDS page. But in some case, if you want to collect those membrane protein and measure their enzyme activity, don't use ionic detergent. You need to use a non-ionic detergent. You need to keep the original protein structure and keep their enzyme activity. Otherwise, you cannot graduate, right? Yeah. Okay? Okay, so if you have, during those membrane protein extraction, if the concentration of your detergent above a certain level, we call the critical micelle concentration, they can form such a, the membrane protein contain the micelle, which usually can maintain the better structure of those membrane protein because they still have a minima, at this minima membrane structure. But if the concentration below those are critical micelle concentration, they only will kind of, you still can extract those membrane protein, but it just have a few lipid aligned on the surface of those proteins. Sometimes it may damage or change the structure of your protein. It may, okay, yeah. So some people care about this critical micelle concentration, some people don't, yeah. Okay, so very quickly, we just introduced we introduced about the GPI anchor as dilation preneulation for the lipid anchor protein. And we introduced about the peripheral protein, which use a phospholipase. It has a tunnel and can extract the phospholipid and digest the chemical bond, use a calcium bind, attract the polar head, and use the hydrophobic amino acid on the wall of the tunnel to keep them inside the tunnel and digest them, okay? And we also talk about integrate or transmembrane protein. So common structure will be the transmembrane domain, it's alpha helix and the non-polar amino acid, but it always have some exception is beta strand barrel. For example, the proline protein and the polar amino acid T cell receptor. Okay, and we also talk about detergent. They are all hydroamphipathic, but the ionic detergent has a charge. They will disrupt membrane, but it will denature. This is denature. With denature protein. Non-ionic detergent, they don't have charge, but they will disrupt the membrane protein, still disrupt membrane protein, but they don't denature protein. Did I go too fast? Or is it okay? Okay, yeah. Okay, so any questions so far? If you have questions, you can even ask in Mandarin. Yeah. Okay? Okay, so next question, we talk about lipid synthesis. Okay. As I mentioned before, ER and the Golgi, or ER is the major organelle to synthesize the lipid. Okay. So why, when we mention this definition, certain things are mainly synthesized in certain organelle. Why? There's only one reason, because the critical protein do the protein synthesis only locate in the ER. That is why this thing only happen to the ER. 99% of this event could be during this reason. Okay, when I claim this thing only happened to the Golgi because the enzyme only exists there. Okay? Okay, it's also the case in the ER. There's a critical protein required for the lipid synthesis only exists in the ER membrane. That is why ER are the major guides to synthesize the lipid. Okay, those are the critical protein in the ER. So, but the lipid synthesis is a multiple step reaction. In the beginning, it's happened to the cytosol. Okay, so acetyl-CoA, which is a important metabolized intermediates, come from, for example, glycolysis. We eat food, and we are in digesting system. We digest food, release the sugar, and the sugar go into the cell. Then we will go through the glycolysis. It will become the pyruvate, and finally become then become the acetyl-CoA. Okay, so acetyl-CoA in the cytosol with some cytosolic enzyme, they will become the fatty acid. Okay, and the fatty acid will conjugate with the fatty acid CoA. Then the fatty acid CoA will kind of use the GPAT and the LPAAT, which is the acetyl transferase. We conjugate with those glycerol phosphates with those fatty acetyl. Then it becomes the really original form of those phospholipid. We call it a phosphatidic acid. Then it still have a phosphate group. Then phosphatase will take out those phosphate group and become diacetyl glycerol. Then those choline phosphotransferase will kind of form the phosphatidylcholine. But actually, we have mentioned before, the PC are abundant in the exoplasmic leaf left or the lumen leaf left. Okay, not in the leaf left. So actually, there's an enzyme determine their asymmetry is a flippase. Okay, they can help lipid flipping. So this flippase use energy to flip the PC from this side to the other side. Okay, so this is the guy determine the lipid asymmetry. Okay, so then the PC will always contain here, and after it's budding out the vesicle and fused with the plasma membrane, the PC will be abundant on the surface lipid leaflet. Okay, yeah. And then you may ask, those fatty acid, they are highly hydrophobic, right? But in the cytosol, it's a hydrophilic environment. How such a hydrophobic stuff can freely and happy in the hydrophilic environment? That's weird. So they need the help for some protein, for example, the fatty acid binding protein. Okay, so this is the soluble protein. It's hydrophilic on the surface. Okay, but it has a pocket right here, which is highly hydrophobic, and it can absorb those fatty acid in the pocket. And the whole complex basically are hydrophilic on the surface, so they can really happy and freely diffuse in the cytosol, and it go everywhere to touch the ER membrane and do such a reaction. Okay, otherwise the fatty acid will accumulate to a certain part that it cannot freely diffuse in the cytosol. Do you know why the vesicle, why cell need to deliver stuff through the vesicle? Or do you know why cell need to deliver vesicle through energy? You know what I'm asking? A vesicle trafficking need energy, right? Why it need the energy? There are several major transport strategies cell can deliver the cargo. First is diffusion. You just synthesize the protein or synthesize molecule, they can passive diffuse in the cytosol. Okay? But why they still need to use a vesicle? The one factor is size. Okay? Actually, the cytosol is a really crowded environment. They are full of a lot of different cytoskeleton, a lot of protein, a lot of different stuff. People have been measured the protein bigger than 20 nanometer will be stick in the cytosol. They cannot freely diffuse. Okay, so if the molecule bigger than 20 nanometer in diameters, they need to use another system to deliver. So the diameter of the vesicle usually range from the 30 to 60 nanometer. So the cell will collect those huge cargo in the vesicle, then budding out from the ER, and they use a motor protein. And you can think about if this cytosol pool is a 20 nanometer, but the vesicle is like a 30 or 60. You need to have energy that the motor protein will use energy to pull and take out those crowded environment and move the stuff to their destination. Okay, yeah. Okay, so next will be talk about this a little bit more detail of this lipid synthesis. So as I mentioned before, when the ER synthesize too much lipid, they will form the lipid droplet. Okay, so those are different steps of the lipid droplet. The first, they will accumulate too much excess, the cholesterol or triglyceride, to the space between the two lipid leaflet. Okay, then they will gradually form such a intermediate structure, budding out, then form the micelle, which is a lipid droplet. How can they accumulate lipid here, and how can they budding out to form the lipid droplet? That has been well studied. Okay, so this is really important because if you accumulate too much triglyceride, you will get a lot of heart diseases and a lot of different homeostasis or diseases. Yeah, right. So people think about if we can find a critical protein required to synthesize the triglyceride or cholesterol, we may generate there. We know the therapeutic target. We just generate the inhibitor of those critical protein. Then we can block our body to synthesize triglyceride. And everyone can eat a lot of wonderful food, garbage food, but we still keep our triglyceride level low, and we can keep healthy. Yeah, that's the perfect situation, right? That's the motivation of our research. Okay, so that is why a lot of researchers they try to screen what kind of they use the yeast cell, 酵母菌, as their model organism, and just feed those yeast cells with a lot of lipid. And then naturally, they will form a lot of lipid droplet. They can easily observe under microscopy. Then they kind of screen, just knock a gene one by one, knock down gene one by one, and start to screen which gene knockout will suppress those lipid droplet formation. And finally, they found several genes are really important, which is the DG81 and the DG82, which are also ER membrane protein. Okay, and those are really kind of therapeutic target people try to find out the inhibitor to make our triglyceride level lower. Okay, yeah. And why? And after several decade studies, they gradually know why they will put the accumulated lipid to the space between the two lipid leaflets. Because there's a DG81, this is a DG81, the protein complex. They have a lateral pocket in this protein structure. So they will synthesize protein, and the lateral pocket will accumulate triglyceride to this end. Okay, that is why they will keep accumulate a lot of lipid or triglyceride here gradually and increase there. Okay, that's because their protein structure. That's a really beautiful story. Yeah. Then next, why the cell can form such a budding out such a lane structure and finally form the lipid droplet? That's regulated by the CPM. Okay, CPM protein. CPM protein is also ER member protein. They can form the oligomer. Okay, oligomer is mean a lot of CPM they work together. They can form the really huge CPM group and work together. Okay, so this is a part of the CPM. Only half of the CPM protein is the ER membrane. So those purple part of the CPM, they are lipid binding domain. They will accumulate the triglyceride or the cholesterol synthesized from the DG81. Okay, they will kind of collect them together, and this is a four CPM protein. You will see there are a lot of transmembrane domain. Those transmembrane domain will bind with those ER membrane. Okay, then they can form such a cluster and form the oligomer, and then finally, from the top view, it looks like this. So they kind of will gradually align with the lower membrane protein and gradually budding out, and also collect the lipid in the core of the region. So very, very beautiful design, right? So after that, they will kind of budding out the lipid like this and form this. Okay, there will be a lot of CPM look like here. And finally, they can form the mature and the takeout from those ER. Okay, so structure of those. So this is an example how DG81, DG82, and CPN can form such a lipid droplet. Okay, next we talk about the cholesterol. So this is the synthesized pathway of the cholesterol. Okay, and this HNG-CoA reductase is the step. This step is a key rate controlling step. Okay, and finally, it will from the acetyl-CoA, it finally will generate a cholesterol. We need to have a proper level of the cholesterol. We don't want too much or too less cholesterol. Okay, but usually we get too much cholesterol. Yeah, so people also try to find if you eat too much, if your body have too much cholesterol, first they will accumulate to this is a section of the artery wall. So too much cholesterol will make those artery wall become the inflammation. So those inflammation will make this artery wall thicker and thicker. Then this structure in this part will become really weak, and then just break down. Then the blood will kind of leak out from the blood flow. So to prevent this, our body will form a lot of platelet, accumulate a lot of platelet here, and form the thrombus to prevent the blood leaking. Okay, but such a big, we call it thrombus, 血栓 will block the blood flow. So the blood flow cannot deliver the nutrient anymore, and we will have a heart attack. Okay, so the one way to cure or treat those we call it atherosclerosis, 动脉硬化, is try to lower your cholesterol level. Okay, so to find out, because people find out this is the key rate controlling step of the cholesterol synthesis. So those are the perfect drug target, therapeutic target. Okay, so it has been found that statin, which is an inhibitor of the HNG-CoA reductase. So this is the popular drug people now use to treat atherosclerosis. It can really lower our cholesterol synthesis, lower our cholesterol level, and make improve the atherosclerosis symptoms. Okay. Okay. So just then, for those lipid stuff, they synthesize. They need to, and they also need to sometimes go to the other part of the cell. How can they kind of transport in this cytosolic environment? They basically through these three mechanisms. The first, they come from here and they can budding out as a vesicle, then come with those vesicle and then fuse with the destination target organelle or target plasma membrane or target site, then go there. Okay, or in some case the different organelle they will interact with each other and they form such a physical contact, and a certain protein will help them to facilitate the lipid mobility and also facilitate the lipid exchange. Okay. Or some binding protein, like I mentioned before, fatty acid binding protein, they have a pocket that can absorb, can take a fatty acid and they have a hydrophilic surface, and they can take those lipid to the other side. Okay, basically you just cannot expose those lipid directly in the cytosol. It's not an energetically favorable situation. You need to use this mechanism, or need some protein help from the protein to do this job. Okay. Okay. So in this section, we talk about the multiple synthesis step for the fatty acid or phospholipid synthesis. Okay. And the flip path is the guys determine the lipid asymmetry. Okay. And the fatty acid binding protein FABPs will bind to the fatty acid and also help them to transport to the destination. And the HNG-CoA reductase is a rate controlling step of the cholesterol synthesis. So this is the therapeutic target. That is the beauty of why we do the basic research. Okay, if you find out those mechanisms, you will know which guys will be your first therapeutic target. And you only need to eat one kind of drug. You don't need to eat multiple drugs. Okay, yeah, then you can achieve your therapeutic purpose. Okay, and we also talk about the three mechanisms of the lipid transport. First is you can use a vesicle transport. Second, you can use the integrated protein facilitated lipid movement between the membrane if they form the physical contact. And the third, several small protein can transport lipid, so you can, with that help, fatty acid can go to their destination. Okay. Any questions so far? No? Okay. So if not, we just move to the next chapter. Okay. Now we talk about the molecule, how molecule transport across this membrane. Transmembrane transport of ion and the small molecules. Okay, so again, we go back to this slide. So the molecule need to transport, the plasma membrane is a permeable barrier. Some guys can go across the membrane, some guys cannot, okay? So we call this semi-permeable barrier, okay? Because the molecule sometimes can go in, sometimes can go out, but some of them cannot. That is why it can accumulate specific molecule inside the cell to generate to perform the living reaction. Okay, yeah. So now we only consider the pure phospholipid bilayer. We don't have any protein, just pure chemical property of the pure phospholipid bilayer. What kind of molecule can easily go across or can permeable? We call it permeable, the phospholipid. What stuff cannot, okay? So the pure phospholipid bilayer are impermeable to the large charge and the hydrophilic molecule because it's easy to understand why the hydrophilic cannot, because those are hydrophobic layer, right? So the hydrophilic don't like the hydrophobic load, so they cannot easily cross those hydrophobic layer. And also charge cannot. And there are some space between those phospholipids, so if the small molecule still can easily go across those space and across the membrane, but the large molecule cannot. Okay, so for example, the gases CO2, N2, and the O2, they can easily permeable for across the those phospholipid. So you can imagine there's no different concentration. The level of the oxygen, CO2 level outside the cell and inside the cell, because our membrane cannot stop them, okay? It's easy to kind of across the membrane. Okay, and those uncharged and the polar molecule like ethanol can permeable water, they slightly permeable urea, okay? And the large uncharged polar molecule like glucose and fructose, they cannot, they need. Okay, and the ion, they are charged, they have a different charge, they are impermeable, okay? And of course, some amino acid, ATP, blah, 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 those stuff are impermeable, okay? And several factors for the movement, just quickly remind you the passive diffusion. So passive diffusion is the diffusion of the substrate across the membrane with no energy investment. There's no energy need to run this reaction. So it's a tendency for the molecule to spread out evenly into the available space, okay, available space. So usually you will transport from the pool with a higher concentration to the one with a lower concentration, okay, until there's no net direction movement. We call it equilibrant, okay. They can evenly distribute to the available space. We call it equilibrant, okay. And such a diffusion. Now we talk about the diffusion, okay? Such a diffusion rate depends on several factors. First, whether those molecules can cross the membrane or diffuse across the membrane quickly or slowly, it depends on those factors. First is hydrophobicity. If this molecule is more hydrophobic, it can diffuse quickly. As more smaller, it can diffuse quicker, okay? Faster, okay? And also the concentration of the gradient across the bilayer, okay? If the molecule have a really high concentration here and really low concentration here, we call this a chemical gradient. It can diffuse more quickly from this side to the other side. So the difference determines the diffusion rate, okay? Not only chemical concentration, the charge also determines this. If the molecule with the charge have a really strong positive charge here, really strong negative charge here, it will also come from one side to the other side more quickly, okay? So we call this, electrical potential across the bilayer or the concentration gradient across the bilayer They determine their diffusion rate. And usually, we will consider both factors together. So we call it electrochemical gradient. Okay? Here, that will kind of determine how fast the molecule can move from one side to the other side. But you can imagine if our membrane only kind of go through this permeability or select the molecule through the pure phospholipid. After a while, they will all achieve the equilibrium, right? So the oxygen will go in and go out, but anything impermeable, they either will always retain in the membrane or always keep it in the extracellular space. They cannot generate specific environment in the cytosol, right? So those membrane need the help with some guys to collect the specific impermeable molecule across the membrane, or release, or kick them out, then generate specific environment. Those guys, helpers, are the membrane protein. Okay, you cannot just simply go through this by the phospholipid. It cannot. Okay, you need to use some protein to specifically collect or take out some molecule. Otherwise, you cannot generate a specific environment inside the cell. Okay, so those are the several kinds of protein can do this job. Let us take a 5-minute break, and then we will introduce those proteins.